Hello class, and thanks for watching our laboratory video all about wine. This video will focus on winemaking and, most importantly, the microorganisms responsible for producing this drink. To start off, let's talk a little bit about the process grapes undergo to become wine.
The alcohol found in ancient beverages was a result of naturally occurring yeast fermenting the sugars found in the foods. When early humans ate or drank the fermented foods, they noticed the change that fermentation caused and began fermenting foods intentionally. In 1876, Louis Pasteur demonstrated the involvement of living organisms in fermentation. In 1883, Emil C. Hansen isolated brewing yeast and propagated the culture. This led to the discovery of the importance of yeast in brewing, especially the use of single cultures rather than mixed cultures. Winemaking developed into an industry. Naturally occurring yeast were replaced with strains developed specifically to enhance the fermentation process and the flavor of the end product. Some wineries and breweries, especially in Belgium, however, use exclusively naturally occurring yeasts that have taken up residence in the equipment, buildings, and vineyards. Fermentation is an anaerobic catabolism of a complex organic compound that results in the formation of ATP by substrate-level phosphorylation. It occurs in bacteria, fungi, protists, and animal muscle cells in the body, producing lactic acid and lactate, carbon dioxide, and water. These waste products are excreted from the cell. In yeast and most plant cells, fermentation produces ethyl alcohol, carbon dioxide, and water. Fermentation oxidizes NADH to NAD+, so it can be reused in glycolysis. NADH is the reduced form of NAD+, and NAD+ is the oxidized form of NADH. In yeast, the overall process of fermentation involves converting glucose to alcohol and carbon dioxide. The reaction occurs using enzymes in the yeast cell. Ethanolic fermentation is used in the production of beer, wine, and bread. Saccharomyces species are responsible for the vast majority of alcohol fermentation. Saccharomyces is a single-cell fungi found living on plant material including fruits, vegetables, and grains. They are saprophytic, that is, they process dead or decayed organic matter. They are characterized by their inability to use nitrates and their ability to ferment various carbohydrates. Saccharomyces cerevisiae is used in making wine, bread, and beer.
Our lab experiment sought to identify the characteristics that yeast imparts on alcoholic fermentations. We looked primarily at alcohol content and pH differences in wine of 7 different yeasts. We chose wine for a couple different reasons. First, we all like wine and wanted to understand the science behind making it. Second, we chose wine over beer because wine is significantly easier to make with less room for error and less raw material costs. There are no boiling steps or hops or grain. We simply put all of the ingredients in a bucket and let it sit for a little bit. Third, equipment from Grace Isaac's microbiology laboratory class was available to us as our students do a very similar project. The only thing that we had to purchase was grape juice and yeast. We followed a recipe given to us by Grace to make this wine, and kept the recipe the same for all of the different yeasts. As all of you know, there is quite a wide assortment of yeasts on the market. We went to How Do You Brew, about 10 minutes from campus, to purchase our yeasts and ask for their recommendations. We chose 4 Red Star and 3 Lalvin varieties to test. The 4 Morningstar were chosen without any prior research, but the 3 Lalvin varieties chosen were based on the yeast's alcohol tolerance. We hypothesized that when our fermentations were complete, we would see clear and distinct differences in the alcohol content. The yeast theoretically would die off as the alcohol content of the wine approached that yeast's tolerance. The yeast that we selected had alcohol tolerances between 14 and 18%. Our recipe called for many other ingredients besides just the grape juice. We added a Campden tablet, which is a sulfur-based product to sterilize the wine and keep it free of any unwanted bacteria and yeast that might have been introduced. Next in our recipe was 3/4 of a teaspoon of an acid blend of citric, malic, and tartaric acids. Looking back, we likely didn't need this, and we will get back to that in a little bit. We then added 5 drops of pectic enzymes. This pectinase is utilized in order to break down pectin and avoid any haziness in our final product. Although this enzyme is in grapes, it isn't at high enough concentrations in order to achieve the desired clarity and effect. The next ingredient is sugar. We all know why this is important. Without the extra sugar, we wouldn't have as much alcohol produced if we just had the grape juice alone. This mix was left to sit for a day in our sanitized fermenters before pitching the yeast. Alright, so now comes the most important part of the wine, which is the yeast pitching. So what we have is 7 different yeasts for our 7 different wines. We're using 4 Red Star yeasts and 3 of the Lallivan yeasts. All of these yeasts have different alcohol tolerances, so that's why we are using them. So what we're going to do is we're going to add a packet to some warm sugar water, and we are going to let it sit for about 15 minutes. This helps to activate the yeast to get it ready for when we pitch it in just a couple of minutes. We also have here some bentonite, and what this does is it works as a clarifier for the wine, and we will be adding that when we pitch the yeast as well. So right now I am adding the bentonite to our wine bases. If you want to come over and take a look at this. This is our inoculated grape juice. It has the original grape juice, about a gallon of it, with sugar and acid tablets and pectic enzymes to help it all move along, and a Camden tablet as well. On top of all of these are the airlocks that we're using to ensure that it's an anaerobic environment. These will let gas bubbles that the yeast produce to escape, while keeping air from going back in. So once we get these yeast pitched, they will be in their anaerobic environment and happy and ready to produce the alcohol that we're looking for. Alright, so now our yeast have been sitting in the sugar water for about 15 minutes, so we're going to go ahead and start pitching them. Bloop! There they go. We let the wine sit for around 2.5 weeks to ensure complete fermentation and that the yeast had reached their alcohol tolerance. Normally in homebrewing of wine, 70% of alcohol is produced in the primary fermentation vessel over the period of a couple days. The remaining 30% of the total alcohol content of the completed wine is produced in a secondary fermenter, like a glass carboy, over 1 or 2 weeks. This is done to avoid any off flavors from the dead yeast due to the initial and rapidly rising alcohol content. In our experiment, we ended up not following this practice to avoid any error from our part as extremely novice winemakers. Our primary goal was to see the differences in alcohol content after the fermentation process was complete, not to produce a particularly tasty wine, although that certainly would have been very nice. Keynote, would have. When it came time to open up the fermenters and analyze the wine for alcohol content and pH, our first notice was the complete lack of bubbles coming out of the airlocks. This was expected and initially very promising to us, as this indicated that the fermentations were fully complete. We suspected that the yeast had reached their alcohol tolerance and all of them simply died off. What we found was particularly surprising. All of the 7 varieties of yeast had produced wine with roughly the same alcohol percentage and pH, and this wine was very unpleasant from a sensory standpoint. All of our wines were a whopping 18%, according to the hydrometer readings. This is certainly on the higher end of the spectrum for wines, and they all exhibited a pH of around 3. Red wines typically are in the range of 3.3 to 3.6, so this was particularly surprising to us to find this high hydronium concentration. We will admit that the strips we used were not sensitive to the 10th decimal place, so take our pH values with a grain of salt. But these 2 factors combined made for some really unpleasant tasting wine. Our first thought when analyzing the data is simply, why did this happen? Wasn't the yeast supposed to exhibit different alcohol tolerances? Weren't we supposed to get different numbers here? Something wasn't adding up. Here's our theories on what happened. The first is our error. We did not properly use our hydrometers. What's supposed to happen is we're supposed to take a specific gravity measurement before the yeast is pitched and after the fermentation is complete. The comparison of these two numbers will then give you your alcohol content. Instead, we relied on a measurement given to us in the original recipe. This should have been re-verified on our own, and we probably would have had a more accurate number on what the true alcohol content of these wines were. But that still doesn't explain why all of the wine turned out to be the same alcohol content. We think that the abundance of oxygen in our buckets caused the yeast to grow very rapidly initially. We had only about a gallon of wine in a 2-gallon bucket, so 50% headspace. We think that because the yeast were able to and allowed to grow so quickly, it caused a lot of alcohol to be produced all at once. And so then the yeast, therefore, didn't really have a chance to die off. Here's the next part of it. We think to solve this problem, there's a couple things that we would have done differently. First, we would have doubled the amount of wine made in that bucket. We think that by limiting the amount of headspace, we would have been able to slow down the fermentation and therefore cause us to more closely monitor and more accurately measure what's really going on and how we can compare that to what's actually happening in home brewing methods. This also serves another purpose because our yeast packets were good for between 1 and 5 gallons of wine. We think if we upped the amount of wine and got it out at that low tolerance for that yeast, we would have been able to also slow down the fermentation that way as well. The second can be explained by S-curves. As I'm sure all of you know, the way yeast produce alcohol resembles an S-curve. It's going to be very high initially, and then it's going to taper off at a certain point. Ideally, their alcohol tolerances, but provided the conditions that we had, that wasn't going to happen anyways. But we think if we had the 2 gallons of wine to start with and were able to slow down the fermentation, we think we would have seen a clear difference in the alcohol content. Here's why. We think the higher alcohol-tolerant yeasts would produce alcohol more quickly, as represented by the green line, and that our lower alcohol-tolerant yeasts would produce that alcohol much more slowly. If we cut this off at a certain point, we think we would have seen 2 different values, or in our case 7 different values, of the alcohol content of the wine. I would think that after that 3 to 5 day mark, where roughly 70% of the total alcohol is produced, if we had stopped it there and how most home brewers, they transfer the wine to a secondary fermenter at that point to get rid of any off flavors that the dead yeast provide. If we had stopped it there, just stopped the experiment outright at that 70% alcohol made mark, like here, we would have seen those different alcohol contents. Instead, we let it go all the way over there for 2.5 weeks, so we were doomed from the start by letting it go for that long. So we would have stopped the fermentation sooner, we would have let the fermentation go slower, and then that's when we think we could have seen the true alcohol limits of these yeasts. Although we had to throw out all of this wine due to its sheer awfulness, we did learn a lot about winemaking and biotechnology and how yeast work in general, and in the end, that is really what matters. Let's now discuss some issues and practices happening in commercial winemaking today. Stuck fermentations can happen to any winemaker, especially novice ones like ourselves. Handling this issue is something that all winemakers dread, and therefore it is especially important to be aware of it.
A stuck fermentation occurs when fermentation has stopped before all the available sugar in the wine has been converted to alcohol and carbon dioxide. Sugar levels are important in a finished wine because of the effect on the flavor and body of the wine, and also on microbial stability. To solve the problem, first be sure that the fermentation has stopped. Measure the specific gravity over several days to be sure it does not change. Check that the airlock is firmly seated in the bung and the bung is correctly attached to the fermenter. ensure the lid of the fermenter is on properly. Gas may still be escaping through a space other than the airlock. If fermentation slows down or stops, it is usually due to some change in the fermentation conditions. Determine the pH, alcohol content, amount of residual sugars, titratable acidity, and total N-free sulfur dioxide. Knowing these results may allow for adjustments to the must or wine. The easiest solution to a stuck fermentation is to finish the wine as is, with the finished product sweet rather than dry. Another option for restarting fermentation is to dilute the stuck wine with a wine that is similar to the envisioned end product. Malolactic fermentation, or MLF, is the fermentation of malic acid to lactic acid by lactic acid bacteria, or LAB. MLF deacidifies the wine, creating a rounder, fuller mouthfeel in the wine. MLF usually occurs after the primary fermentation has ended. Most red wines that will be aged and a few white wines undergo MLF. The main advantage to MLF is to decrease the sourness of the wine, and also it uses the remaining nutrients in the wine, decreasing the likelihood of microbial contamination. As with yeast fermentation, it is highly likely to occur unless prevented. All wine LAB are gram-positive, non-mobile, and non-sporulating. They are facultative anaerobes that require a rich medium. Optimum growth temperature is between 20 to 30 degrees Celsius. Lactic acid bacteria occur naturally on grape leaves and stems. However, most winemakers inoculate with a selected strain of bacteria such as Onychococcus oeneus. The specific strain selection is based on temperature, pH, alcohol tolerance, sulfur dioxide tolerance, and type of wine desired. The alcohol levels at the end of primary fermentation may be high enough to inhibit bacterial growth. But with proper nutrition and environmental conditions, ML bacteria can usually adapt its growing conditions. If the temperature of the wine becomes too warm, then the bacteria are more likely to feel the effects of alcohol. If the wine becomes too cool, then reproduction and fermentation will decrease. Typical pH range for survival of ML bacteria is between 3.1 and 3.6. However, there are bacterial strains that can survive outside of this range. If the pH of the wine is too high, then while bacteria have an easier time thriving, the wine becomes more susceptible to contamination by other bacteria. High levels of free and bound sulfur dioxide can inhibit the growth of ML bacteria. An acceptable range is between 0 to 10 parts per million of free sulfur dioxide and 0 to 30 parts per million for total sulfur dioxide. In general, adding 1 Campden tablet before primary fermentation will keep the sulfite level within acceptable range by the end of primary fermentation.
The future of the wine industry may look to genetically modified organisms to meet consumer demands. Genetically modified yeast may be used in wine production fairly soon. Saccharomyces cerevisiae, the yeast used in the fermentation of wine, has evolved a highly efficient strategy for energy generation which maximizes ATP energy production from sugar. This adaptation enables efficient energy generation under anaerobic conditions and limits competition from other microorganisms by producing toxic metabolites such as ethanol and carbon dioxide. The yeast's fermentative and flavor capacity forms the biotechnological basis of a wide range of alcohol-containing beverages such as wine. The ethanol content in wine has increased as a result of consumer demand for improved flavor. Recently, a trend has emerged toward lowering the ethanol content in alcoholic beverages. One way to decrease ethanol concentration in wine is to use yeast strains that have been genetically modified to be able to divert some carbon away from the ethanol production. These yeast strains have been created based on a large number of gene modifications that have been known to generate an impact on ethanol formation. According to one study, which assessed 41 modified yeast strains, enhancing glycerol production by increasing expression of the glyceraldehyde 3-phosphate dehydrogenase gene, GPD1, was the most efficient strategy to lower ethanol concentration. While this was generally successful, additional modifications were still needed to avoid negative effects on the wine's quality. The results of this study showed that the strain AWRI 2531 was able to decrease ethanol concentrations from 15.6% to 13.2%, whereas the strain AWRI 2532 lowered ethanol content from 15.6% to 12% in both Chardonnay and Cabernet Sauvignon juices. Both of these strains produced high concentrations of acetaldehyde and acetone, which negatively affect wine flavor. The further modification of these strains allowed the concentration of the metabolites to be reduced. The process of creating ethanol-reducing yeast strains is rather complex. The first step in creating these genetically modified yeast strains is cloning the GOX gene, which encodes glucose oxidase. This gene is amplified by PCR from genomic DNA of Aspergillus niger and cloned into a plasmid that encodes the secretion signal from the mating pheromone factor MF-alpha immediately downstream of the FBA1 promoter. In the resulting plasmid, the GOX gene was cloned in frame with the MF-alpha secretion signal to enable secretion of glucose oxidase to the extracellular medium. The next step is the determination of glucose oxidase activity. The yeast colonies transformed with the PCR product containing the MF-alpha secretion signal GOX gene construct were screened for their ability to secrete active glucose oxidase using a plate assay. The colonies on plates were briefly overlaid with 10 milliliters of 0.1 molar K2HPO4 buffer with a pH of 7, containing 10 grams per liter glucose, 1% agarose, 100 milligrams per liter dianisidine dihydrochloride, and 15 units per milliliter peroxidase. These plates were then incubated at 37 degrees Celsius for 1 hour once the overlay agar was set. Colonies secreting active glucose oxidase produced a brown halo. Once the desired strains are selected, they are evaluated for decreased ethanol production in YPD10 medium. Aerobic conditions were attained by growing cultures in flasks covered with aluminum foil that allowed a free gas exchange with the environment. Cultures were fermented at 28 degrees Celsius for 48 hours. At the end of fermentation, samples were collected and centrifuged for 5 minutes at 15,000 G, and the cell-free supernatants were kept at 4 degrees Celsius for high-performance liquid chromatography analysis. Strains showing a considerable reduction in ethanol concentration after fermentation were assessed for their ability to ferment chemically defined grape juice medium and/or grape juice. Yeast growth was monitored spectrophotometrically by measuring absorbance at 600 nanometers. The results of this study were based on the genetically modified yeast's ability to reduce ethanol content in the final wine product. To determine the effectiveness of each new yeast strain, the yeast must be screened for gene modifications that impact ethanol production. A collection of mutant S. cerevisiae wine yeast strains were generated from the same parent, AWRI 1631, to identify the best candidate genes for constructing low ethanol strains. Expression of genes encoding enzymes that divert carbon away from ethanol production was increased, whereas genes that contribute to ethanol formation were deleted or downregulated. Target genes included some involved in glycolysis, pentose phosphate pathway, and TCA cycle, some regulating glucose repression, and some with the potential to catabolize glucose in the medium. Characterization of low ethanol strains under winemaking conditions in chemically defined grape juice medium must also be taken into consideration. 4 strains selected from the above screening were assessed for their ability to ferment chemically defined grape juice medium in winemaking conditions. Only one strain, AWRI 2532, was not able to complete fermentation, as was evident from the high concentration of residual sugar in the medium. All 4 strains produced fermentation products with higher glycerol and lower ethanol concentrations than the parent strain. One strain, AWRI 1631 ASPTPI1, produced more malic acid, acetic acid, and glycerol with less succinic acid and a 2% lower ethanol concentration than the parental strain. Compared to the parent strain, AWRI 1631 3GPD1 produced slightly less malic acid and succinate acid, 6 times more glycerol, 20 times more acetic acid, and 20% less ethanol. AWRI 2531 produced less malic acid, slightly more succinic acid, 6.1 times more glycerol, 3 times more acetic acid, and 22% less ethanol. Today, there is a growing interest in alcoholic beverage industries to reduce the level of alcohol in beers and wines. There are many strategies available to achieve this end goal, but all have serious limitations. It is widely believed that microbiological approaches have the capacity to deliver the best outcome. A large number of target S-cerevisiae genes have been manipulated to test their potential for the development of a low ethanol wine yeast strain. Of all the strategies aimed at decreasing ethanol production, those intended to increase glycerol formation were the most effective. The efficiencies of several strategies that have been shown to alter ethanol formation, including diverting sugar to lactate production, manipulating hexose transporter genes, and expressing an H2O forming NADH oxidase, as well as novel strategies such as diverting carbon to the formation of reserve carbohydrates, remain to be tested.